So good morning, everyone, and thank you for coming. Also, thank you for those that are online. Today we have a talk by Enrico.rezende, Thermal Tolerance Limits in a Warming World. Enrico is an ecologist, an evolutionary biologist. He's from Brazil and Chile, and did his PhD at University of San Diego Riverside. Then he came to Spain, worked in Barcelona as a Ramón y Cajal, and then we lost him to Chile. So he's now associate professor at the Pontifical Catholic University of Chile. Maybe he will come back one day. And today he's one of these ecologists, call them integrative ecologists, trying to address questions in ecology from several angles. Of course, evolution and ecology. He's also known for his work in thermal physiology, and I guess that's what he's going to tell us today. Yeah, so the floor is yours, Enrico. Thank you.
Hello, guys. Thank you so much for having me here. Miguel, thank you for the invitation. It's a pleasure to come back to Spain. I haven't been here for the past 13 years. And so I'm going to be talking about thermal tolerance in a warming world. And as Miguel said, so just to put things in perspective, there was this book by Michael Anguleda where he was talking about thermal adaptation, and essentially he mentions that biologists have described patterns and documented effects to the extent that empirical knowledge, data that we compile, has far outstriped theoretical understanding. So the main point here, and that's from 2009, is the fact that as thermobiologists, we have a lot of data, we have accumulated tons of data, but we don't have much of a theoretical understanding of how to use this data for predictive purposes. And essentially, that's how I started my work in this area. So this is a very famous paper by Curtis Deutsch. And for those that don't know, he's going to be here in Granada now on Monday. So we have the symposium, so it's an advertisement anyway, so that's the reason why I'm here. And Curtis, what he did with this work was showing that species from tropical environments, they were closer to the tolerance limits than those from high latitudes. And essentially what you had was that the climatological predictions that the global change or climate change was going to be more extreme closer to the poles. It was inverted when you take into consideration the physiological information that we have about different species. And so that means that instead of having more climate change in the poles, the impact of this climate change, the impact of this global warming was going to be higher, or is going to be more pronounced close to the equators. And that was in 2008, and that changed the frame of mind of many of physiologists, myself included, that suddenly we realized that physiological knowledge is extremely important to see or to predict what might be coming in the future as temperatures rise. Now, the problem with that, and that's essentially my contribution to the area, was to realize that there's a problem with what people call critical thermal limits. Essentially, when you talk about animals can tolerate a given temperature, right? What you have underlying that assumption is that you have a threshold model. Essentially, below that particular threshold, the animals can tolerate the temperature. Above that threshold, the animals are going to collapse. And in principle, people would think that, if you measure them in the lab, you can estimate the threshold, and then eventually you can do all sorts of predictions based on that, estimate. And that's actually incorrect. Right? I'm not going to get into too much detail of why, but we're going to see how to circumvent this problem. And aside from being conceptually flawed, you have a problem, and that's how I started working in this field, that we try to measure these critical thermal limits with different measurements or different protocols, and every time we change the protocol, the estimate would change. And so you'd have like a threshold varying from 30 to 35, depending on how you'd measure that. And so eventually the main conclusion is that these methods are actually highly unreliable. And the problem with that, and I'm showing that, for instance, in the panel to your right, is that people are still publishing a lot of, predictions of what might be coming in the future using this sort of limits. And you can see that, even in papers like in Nature, they're still using unreliable estimates to predict what might be coming in the future. And so the main point of the talk nowadays is why this is wrong and how we can circumvent this problem. So in 2014, I published this paper in Functional Ecology, and it was a big criticism of why critical thermal limits didn't work. And to make a long story short, the problem is that when you start increasing the temperature of a water bath, for instance, you have two components that are not being taken into control. One is time, and the second one is temperature. So actually you have two dimensions instead of a single one that will determine how much of a temperature or how much of a stress a given species can tolerate. And so instead of doing that, with the ramping experiment, the way we proposed how to circumvent this problem was actually to measure organisms at constant temperatures. And so let me go through first this left panel here, and then we go into the 3D model. So what you have here is a measurement where you have a bunch of individuals and you start measuring them at time 0. So you put them in a water bath and they're all alive, and then you put them and they start increasing the time at a particular stressful temperature. And so what might be happening, for instance, in the longest curve there at 30 degrees, is that you have everybody surviving, but at a given time you start having animals collapsing. And they start dying, and eventually they reach 0. So you have here the percentage of survival, and at 30 degrees you can see that they drop up to 100% collapse at a given particular amount of time. If you increase now it to 31 degrees, they're going to die sooner, and then 32 and 33 and so forth until 35. The only difference between 35 and 30 is that the animals are actually going to be dying, collapsing out of heat, right, before than what would happen at 30 degrees, okay? So the way we can describe now thermal tolerance, and by that I really mean the description of the variable thermal tolerance, the trait, is it's a 3D model, and that's what you can see in your right. And essentially what you have is a dependent variable, which is survival probability, and it's going to vary as a function of the interaction between temperature and time. And that's why we developed this idea of thermal tolerance landscapes, which you can see over there, which essentially have probability of survival in the z-axis, if you want, and then it's going to be varying with temperature and time there. And so that's the real description of thermal tolerance instead of a critical thermal limit. Okay, so, sorry, there you go. And so one of the important things to keep in mind here is that ideally you want to have some sort of association between what might happen at 30 degrees and 35. So how do you see that? And what we realize is that there is a statistical regularity, and the regularity looks like this. If you plot the temperature versus the logarithm of time, you can see in this case for a 50% mortality or 50% survival, so the average temperature, the average mortality, if you will, and you have a linear relationship between temperature and the logarithm of time. And so this is the 2D projection of what would be the thermal tolerance landscape. Instead of having survival probability as the whole 3D surface, what you have now is for a 50% probability of survival, you have the separation between being alive, which is in white, and being dead, which is in gray. Okay, so now you have a function that's separating life from death. And the function there is exactly the same, okay? And there, so you guys know, for a linear regression, it's very simple to describe this linear regression. Essentially, you have an intercept, which is T max, where we're defining survival following 1 minute of exposure, and you have a slope, which is Z, which is thermal sensitivity. But, an intercept and a slope for a regular regression line. And the first thing that we did in 2014 was checking how general this relationship was. And we found we found in insects, and eventually found it in bivalves, in mollusks and fish, that you could see this relationship being observed in many different animal taxa. So it's in animals. It's been described in bacteria and fungi since the beginning of the 20th century. Actually, many people in the canning industry, they would use the same linear regression to see when you have like a sterilized canned products. And that was during the 19th century. So actually this framework has been employed before by people in microbiology before, us have been employing it. And nowadays we've been able even to generalize it to plants. So it's a very, very general relationship that you see across all organisms, as far as I can tell. And the big question is, does it matter? It's like, is it, does it change the way we perceive the world or the conclusions and the predictions if we actually include time? And this table here, it was published in 2014, and let me go just take you through the table just to show how much it can actually matter, okay? So I replicated a review that had come at that time from Ari Hoffman, and they shown that the upper tolerance limit, so heat tolerance, didn't vary with latitude. Okay, so you have there the CT max, or the thermal limit, has no variation with latitude whatsoever. You can see, for instance, that the lack of any sort of a latitudinal gradient can be seen with the value of the P, right, which is non-significant. So there was no change in heat tolerance between tropical organisms and those from the poles. Now, I replicated the measurement using their data and the R-square, sorry, it's, you know, the explained variance is only 2% of the variation. Now, I replicated their analyses, but now including the logarithm of time, which is supposedly what matters, and you can see that we increase now the variance for 64% of the variation by only including the logarithm of time. And now, if we include latitude following that, you can see that you can find easily the latitudinal climb there. So by the time you control for this confounding factor, which is the amount of time that you're taking to measure these limits, you can also unravel. You can take the latitudinal gradient, and the slope of this gradient, it told us that heat tolerance increases about 0.15 degrees Celsius per degree of latitude. So that means that tropical organisms, organisms at around latitude of 60 can tolerate roughly 9 to 10 degrees more than a tropical organism, okay? So it's a massive effect, and it's an effect that you cannot detect if you're not controlling for time or if you're not essentially explicitly using this type of framework instead of, you know, just the typical critical limits and the threshold model. So far so good. We were able to show that, you know, if you go to the lab and you measure organisms in a constant environment, you can have that, you know, relationship between temperature and the logarithm of time. But one of the problems was that, how do you use that for predictions? And so in 2020, we were able to generalize the model employing differential calculus. So again, let me go through this. So the two panels here, these two, are essentially the same. It's what I showed so far. All right, you have a given temperature, and the organisms, they start dying. So this is many organisms in blue, many in purple, and many in red, and they start dying as temperature goes by. Okay, and you can see here the typical relationship between the temperature and the logarithm of time. Now, a projection of this relationship here can be seen here, right, which was the first slide that I showed you guys, which is the survival probability. It drops. You know, in the cold, it drops slower, but eventually, if it gets very, very warm, in the red curve, you have the survival probability dropping very, very rapidly. Okay, so you have these three curves, which are essentially the projection of this data that you see in here. Okay, so, understood? And so the question is, how do you extrapolate these three measurements obtained at three different temperatures to fluctuating temperatures? And essentially what happens is that you can now, and that's your right panel over there, as you increase the temperature from T1 to T3, what's happening is that the curve, it slowly changes from the blue curve up to the red curve. And if you do that with differential calculus, you can see that the curve looks like it starts as in blue, and then eventually it becomes purple, and at the end of the experiment, it becomes red, okay? That means that as the temperature is increasing, the derivative, the slope now of each of the curves is changing in time as the temperature itself changes. Okay, so that's pure calculus. And even though it's like we consider that, okay, so mathematically it makes sense, we wanted to check whether that worked or not. And so just to summarize, the idea is that you can actually have, with very, very high resolution temperature records, you can transform now the temperature records into instantaneous mortality. Okay, and that's what you see here. The upper panel is the temperatures in Santiago, so with a 1 minute degree of resolution. Okay, so it's daily fluctuations, and if you recall, southern hemisphere, so we have our summer in February, so you start in winter up to summer. And in the bottom panel, you have the daily mortality calculated for Drosophila, for some that we have, like the thermal tolerance estimated in the lab, and this is how it looks. So forget about the two colors. Let's go for instance, for the cold acclimated. So these are flies that are acclimated to 13 degrees Celsius. And you can see that these peaks, the peaks here are peaks of mortality. So eventually, around the beginning of November, you start having that those temperatures over there, they would translate into mortality on a daily basis, right? And eventually it becomes so warm that all the flies would die, all of them would die within that particular thermal regime at around, like, let's say, 30 degrees maximum. Okay, so you have now these instantaneous mortality regimes, and the predictions then, as far as you can tell, right? This is so far it's only the model. Is how you translate a thermal tolerance landscape in combination with the environmental temperatures. These are the predicted mortalities. The question is, how do you validate it? And there are many different ways of validation. So you could contrast the predictions against ramping experiments, the same that I told you with critical thermal limits, right? You just increase the temperatures in a water bath and you see what might come out of it. Or you can do it under, comparing mortality under natural or semi-natural conditions. So this is the typical setup that we use to calculate thermal mortality in the lab, and you can use it with constant temperatures or ramping or increasing the temperatures. And we can also have like mortalities in natural environments. We can detect it and see whether the model holds. And essentially for the past maybe 5 years, one of the things that I've been doing with many colleagues is validating the predictions of the model, making sure that it works. Okay, so let's go one by one here. So for instance, with Drosophila, we had this ramping experiment, and we had 13 different species of Drosophila, and they were exposed to 3 different protocols. They had a slow ramping rate, all right, which is over there, so it's 0.05 degrees Celsius per minute, so it's warming up very slowly, and eventually intermediate rates, so it's 0.1 degrees Celsius per minute, and eventually very rapid rates, 0.25 degrees Celsius per minute, okay? So 3 different protocols. And you can see that the predictions there, so we have the time predicted according to the model in the x-axis, and the time measured in the experiments, and you can see that the predictions work perfectly. Right? I mean, the R-square is 0.999. And instead of, just plotting the time, we can actually see the temperature that the model predicts versus the temperature obtained in the experiments. And again, we have like 0.96% of the explained variation, right? So the model predicts very well. And the dotted line is actually the one-to-one line, x equals y. Right? And so that's one way of validating the model, just against experiments. But we wanted to see, all right, so can we predict what happens in the field, in nature? And the way we did it was we obtained this thermal profile here, right? This one here. And you can imagine that the probability of a given fly to survive for all this period is going to be the probability of surviving the first day times the second day times it's a multiplication, right? It's just probability theory. So we would have the cumulative survival probability curve, which are these ones here. All right, so these are the probability of surviving for a given amount of period, and you would multiply it day after day. And so these are the curves, the theoretical predictions in blue for cold acclimation and in red for warm acclimation. And I think you guys see there that there's a shaded area, right? There's some, shaded gray. So this gray is the field abundance of Drosophila obtained over 7 different years. All right, so in 7 different years, you have this Drosophila subobscura. It would increase in abundance, at the beginning of spring, and then eventually it reaches a given level, which when our model predicts that the population should start collapsing, the empirical population will start collapsing as well. And that happened 7 years in a row. And so the predictions of the model, when we say that, essentially temperature should be an issue for this population, it actually happens to be when we look at the abundances in the field. Okay, so for this particular system, we validated with 2 different ways, and you can see that the predictions of the model for fluctuating temperatures works very well when we compare it against ramping experiments or data in nature. So we started checking, let's see if it works for other systems. And in this case, this just came out, so it's in evolution. I think it's available now in the present, in the current volume. And this is for fish. So we found 2 different data sets. We found one that was published in 1972 with the thermal tolerance curves, the ones that I showed you with intercept and slopes. And so we did the typical analysis to see the variation across these fish. But then there was a second review of critical thermal limits obtained under a variety of different ramping protocols. And so we used one review to predict the output of the other review. Okay, and this is what you see here. All right. And the interesting thing, the 2 reviews, they had very different experimental conditions, thermal acclimation, and so forth. And so even though they were completely different methodologically, we were able to predict from one review the output of the other, as you can see with an R-square in this case of 0.82. Okay? So again, this is the predicted values here in the x-axis and with the results in the y-axis. And again, it works for temperature, and it also works for the predicted amount of time. Okay, so we validated for Drosophila, we validated for fish, for many species. We checked what happens with some crustaceans, some amphipods in riverine systems. And in this case, it's a semi-natural condition, so there was some data predicting, some data in the literature that was showing how many would survive at a given acclimation regime. So again, you have the empirical data now with the gray dots, and the theoretical expectations are with the dotted lines. And once again, the model predicts very well what you'd expect in these, semi-natural conditions. So once again, I mean, you can predict quite well what's happening in different experimental conditions. And finally, another way of validating this, and this is a very neat experiment, we use this capture-recapture approach where we would have this, a freshwater crab, we'd put them in these, small urine vials, okay? And we put them in these small cages, so you had each cage had 20 individuals, and we'd put them in a river in different spots. Some of them were exposed to the sun, they were warmer waters, some of them were, in shaded environments, so it was cool. And with these different engagements, we put data loggers recording the temperature, and that's what you have over there. So you can see that, you have some cages that were in very warm, warm environments and some others that were, in cooler waters. And with this, information, plus the thermal death time curves that we obtained in the lab for this freshwater crab, we predicted how the mortality should happen in the dotted lines down there. And you can see, sure, when it's cool, nobody dies, that's the expectation. But when it's warm, you'd expect, the animals to die at a given rate, which is in the dotted line. And you can see that the experiment, we went every day and collected, we checked how many individuals were dead versus alive, and you can see that the mortality in the field tracked exactly the theoretical predictions that we have. Okay, so you can see that, the model predictions work very well for different organisms under different circumstances that can be validated, either in the field or in the lab. Okay, so the basic idea is that behind all this, we have this proposed paradigm shift, right? We're pushing forward the idea that we should change from the use of critical thermal limits, which was something that was adopted just for logistical purposes. It was easy to measure, okay? But now we realize that it doesn't work that well. And so it was a phenomenological approach. People would assume a static endpoint or a threshold, right? It has very low resolution, and it has very low predictive power. It used to work well when we had, for instance, organisms from different environments, and we wanted to see how their physiology would vary. So you'd compare, God knows, crabs from cold versus, warm environments. You'd see that their critical limits are different. So in that sense, ecological variation would explain their physiological variation. That's fine. It's good enough for that. But the problem is, by the time we have climate change and you want to use physiological knowledge for predictive purposes, then this works very bad, very poorly, okay? And so the idea is that we should change now for a more mechanistic approach. The models have to be dynamic because, stress accumulates over time, probabilistic because it's not that you're either dead or alive. There's a fraction of organisms that might survive and others won't. This is very high resolution, and as far as we can tell, it has a very, very good predictive power. And so the paradigm shift is, even though before we use ecology to explain physiological variation, now we want to, close the switch and essentially use physiological knowledge, physiological understanding to predict what might happen in terms of ecology. Okay, and so the question is, what can we do with that? All right, and at the end of the talk is essentially that. Well, there are many different ways we can use this sort of approach. So one of them is to diagnose vulnerable lineages, okay, those that are more threatened by warming temperatures. And we did this, for instance, with Antarctic organisms. We had this empirical data set here, which was obtained under a variety of conditions, and we were able to reconstruct with some, statistical, as I say, statistical hocus pocus, some very, very sophisticated models. We were able to reconstruct how their thermal landscapes would look like. So these landscapes there were reconstructed from this type of data here. And with that, we would combine it against the temperature recorded in Antarctica nowadays, and we could show that depending on the thermal regime, many of these organisms nowadays, they're already encountering some temperatures that might be stressful for them during summer. Okay, and you can see here, for instance, that in this case, we have chordates being exhibiting more, fish exhibiting more of a mortality than others, for instance, such as echinoderms or mollusks. Okay, so essentially in this case, who lives and who dies will depend on the thermal conditions, of course, but also on the species physiology. And we can use this type of approach to diagnose who might be, more resilient and who might be under threat. Another work, and this was done by Andrew Villeneuve and Ethan White, just as a curiosity. Andrew Villeneuve, he's the cousin of Villeneuve from the director of Dune. Well, that's his cousin over there. And they were simulating heat waves using the same approach to the ones that we developed. And so what you have here is essentially let's say, the background temperatures, and they would simulate these peaks of heat waves with different intensities. So that's the height of the peak and different durations, okay? And they would combine these simulated heat waves with the typical thermal death time curves that we have over there. So you can see, for instance, in this case, it's the peak of heat wave over there, and the mortality in red. You can see how suddenly you have these animals that start dying. And when they did this for heat waves, they're simulating it in different environments, at different times of the year. So the background temperatures are the ones at the bottom, all right? So that's summer now in the north, so summer in August, and eventually winter in December. And they're simulating heat waves at different times of the year in a marine environment. And what you can see here, eventually that's the result of the simulation, is that you might have a heat wave, for instance, during winter, that because it's winter, it's not going to have much of an impact in mortality, but if you have exactly the same heat wave, the same delta when compared to the background temperature, which is in summer, then you might have a massive mortality. Okay? And so they were showing in this paper here that essentially the way climatologists are describing a heat wave is very different as the way that we ecologists should be describing it, because it's not only about the delta in temperature with respect to the background temperature, it also depends on when that heat wave is happening. Okay? And again, this model is allowing us to understand in more resolution this sort of dynamics of mortality associated in this case with heat waves. Okay? So again, the take-home message is that the impact will depend not only on the intensity, but also when it's happening during the year. I mean, it's common knowledge, right? But the thing now is that we can actually predict it with way more accuracy than we could before. You can use this sort of approach to have like long-term predictions regarding the impacts of climate change. So we did this with Wilco Verberg. He's also going to be here in Granada now on Monday. And we did these measurements in amphipods, and these amphipods were in the Rhine River, and we measured the thermal tolerance curves following warm and cold acclimation. And this is how it looks. This is the same thing that I've shown you for Drosophila. So we use the high-resolution temperatures to estimate daily mortalities, which is, the shaded areas here in blue and in red. And eventually, you can have the annual mortality, so which is the cumulative one, the same as Drosophila. You just multiply survival probabilities over the course of the year, and you end up with those data points over there, which is the calculated annual mortality based in high-resolution temperature data, okay? So you can see here we simulated for current environments, and then we just added 1 degree and 2 degrees. We get the temperature profile and increased by 1 degree, by 2 degrees to see what would happen if you simulate this, increase in temperatures. And so you have the current conditions, and then +1 and +2 degrees Celsius. Now, the interesting thing is, by the time you do this, and you correlate these values, the resulting values, against the summer temperatures, the average value of the warmest month, and you use this logistic regression, you can see that the predicted mortality correlates very well, right, with the warmest month. So that could be our proxy to try to infer what would happen with thermal selection. And climatologists, they have these predictions of how these temperatures they should increase in the Rhine River from now up to 2100. So that means that we can simply get this logistic regression here and translate it now into time, and we have some idea of how the amphipods would respond to these different temperatures as the century progresses. Okay, so suddenly we have the possibility of having like better and more highly resolved long-term predictions. We can study geographic distribution limits, right? And so the same thing, the same rationale over there, temperature, and you have the daily mortality and the cumulative mortality. And with that, we had different localities in the US, and we were monitoring, or they were monitoring water conditions, and we used that, and we combined it with envelope models to see where different fish would be located depending on their present distribution. And so what you have here in the open points is the predictions from the envelope model, and what you have with the blue data set is actually our theoretical predictions. And you can see that just by measuring thermal tolerance in different species, you can have a very, very good prediction of when the animals, they find this boundary, right? I mean, they're present over here, and suddenly after above, let's say 20 degrees Celsius, they're no longer present. So you have this inflection point here of the localities where they can survive and those localities where they can't. So you can actually see thermal boundaries in this case. This is how the curves look like. The ones that I showed you before was the one in blue. And in here we have the predictions of the maximum temperature based on this physiological model. And this is the occupancies based on the species envelope models. And just to show how good it is to combine the two approaches. For those species where we detected a limitation by temperature, we have like limited by temperature there, they fall more or less in the 1-to-1 line, which is the dotted line. But of course, many species, they're going to be found in environments nowadays, in distribution limits that we can, they do not encounter warmer temperatures because these temperatures don't exist. But we can predict what would be their tolerance limits based now in physiological data. And so these are the open points over there. You can see that they're inhabiting the warmest localities in the US. So that's all around 30 degrees Celsius over there. And so we wouldn't be able to know, according to envelope models, when they would be threatened by warming temperatures. But now with physiological data, we would know more or less, exactly when these species would be threatened, what they can actually tolerate based on the measurements in the lab. And with that, we can do, for instance, these sort of predictions here of the percentage of suitable habitats as temperature increases. So we have temperature warming here, so these are current conditions in 0, and eventually we can simulate, +1, +2, and so forth. And you can see from these data points, which was the 66 localities that where they could be present, we can see how their suitable habitats can decrease as we're simulating warming temperatures. And then we can see, for instance, which species are more or less threatened based on their thermal tolerance, which is there. And the CT max is the intercept of the TDT curve, and the amount of habitat they would lose on a per degree Celsius basis. As temperature increases 1 degree, their habitat decreases by some amount, okay? So we can use physiological predictions and combine them with envelope models to study how global warming might impact geographic distribution and distribution limits. Okay? We can also study thermal selection in space and time. So we've done this with this intertidal mussel here, snail. And I've been working with some people in Portugal and in the UK, and they have obtained these high-resolution data of intertidal communities here, in this case, from Sagres up to Scotland. And so they provide me the data loggers for shaded environments, which is in gray, and for those environments exposed to the sun, which is in yellow. So you have the temperature fluctuations there on a daily basis and across many years. You can see that it's from 2011 up to 2018. And so with this data set, we simulated whether the snails would be, would fall on a thermal coma or die, which is in orange or in red. And you can see that, in some peaks there during the summer, you'd have many differences across localities and across seasons, of course, on what would be the amount of mortality associated with temperature. And you can see that it varies both in space and in time, okay? We didn't get into too much detail with this, but we know that it's feasible. Another interesting thing in this case here is that all the mortality was detected when the animals were exposed to the sun. In the shade, there was no mortality whatsoever. And what we're working nowadays is we want to generalize this model not only to include temperature, but also to include other factors that would be working synergically with temperature. In this case, for instance, it could be dissolved oxygen. So you have the same freshwater crab that we worked before. So in this case, you have catfish and also gambusia, which is an invasive species in Chile. And you have the thermal death time curves, the same of temperature versus logarithm of time. And you can see now under normoxic conditions, so you have a lot of available oxygen in the water, you can see that these animals tolerate for longer times. But when the water becomes hypoxic, the three species, they exhibit the same type of response, which is they can tolerate less time at a particular temperature, right? So for instance, here in 32 degrees, you can see that the fresh water can tolerate up to 100 minutes. But then when it's a hypoxic environment, in this case, it's 50% dissolved oxygen, it can only tolerate 10 minutes, okay? So what we did is we used this generalized linear model to include the impact of dissolved oxygen on the slope and the intercept of these TDT curves, okay? And what we did then, so we combine now these high-resolution records of temperature with simulated amounts of dissolved oxygen. So there you go, so oxygen concentration there. It affects the slope of the TDT curves, which is Z over there. And with that, we can have the generalized model now to see what happens to mortality as a function of the average temperature, which is the one in black over there, okay? So it's average temperature during the day or of the day and oxygen concentration. And so you can see, for instance, that you have more oxygenation, you can actually tolerate higher temperatures, right? So you have this sort of increase. The blue and the red is computer simulations using simulated data, and then we include all that in a logistic regression, in a multivariate logistic regression, and we can get this type of information there, how thermal tolerance would vary as a function of dissolved oxygen or vice versa, right? How you can tolerate or how you can survive at different oxygen concentration depending on the temperature where you are. And, oh, there you go. Let me see one thing here. Oh, no. So, and essentially an interesting thing is that we can tabulate this. So you can see here, for instance, the shaded areas are those areas that the animals can tolerate the combination of a given temperature and a given oxygen concentration. But we also have as an output a table that tells you the daily mortalities as a function of the temperature and dissolved oxygen. So we're providing it to policymakers or actually monitoring agencies in Chile, and they can actually go with this table and they can measure the temperature of the water and the dissolved oxygen, and they have some idea of whether that particular combination could be lethal or not, depending on the species that are living there, okay? So that means that suddenly you don't need to go and be measuring mortality all the time. You can actually do it in the lab. You have the descriptions of what the animals are doing under laboratory conditions, and then you can go have these tabulations and check in the field whether the water is good quality or not, okay? Another thing that we've realized is that when plant physiologists, they started measuring photosynthetic efficiency, the same relationship between performance and time and temperature holds. Okay, so by now we're generalizing this to plants, and it's just published in Ecology Letters now at the beginning of the year. And essentially what happens, if some of you guys knows about plant physiology, you probably know more than me, but the point is that photosynthetic efficiency, it varies, or the photosystems, they collapse just as we see the animals, they collapse as well with thermal stress. And we were able, again, we combined the empirical measurements with a logistic approach, and eventually we were able to reconstruct what would be for a given threshold. In this case, it's the 50% here, so it's 0.4 of photosynthetic efficiency. You can have now controlling for this 0.4, you have the same sort of association between the temperature and the stress duration, the logarithm of stress duration. And you have the same relationship that we detected for animals. And by the time we published this, another 2 groups in plants, they published the same relationship across many, many plant species. So we know that what we've been doing for animals for the past decade now can be applied for plants as well. So I applied the dynamic model here for plants as well. So you have the temperature of the leaf. Now, in this case, it's adjusted. We have the ambient temperature, and we can do a calibration, right? Depending on how air temperature looks like, you do it against leaf temperature. And with that, we can apply the same model that we use for Drosophila. And you can imagine, you can simulate how photosynthetic efficiency collapses as a function of temperature. Okay, and you can see in this case here, it was the same trees that from the beginning up to the end of the summer, the same summer season, and what's happening is that they're exhibiting less thermal tolerance, most likely because of dehydration. They become less and less hydrated as the summer goes by. Their tolerance decreases, and what you have then there is that the probability of reaching this T50, right, the 50% collapse of the photosystem, it drops as the summer goes by, okay? And so we're collaborating with some people in Australia to see whether, for instance, those massive mortalities that you detect in some woods could be related with this type of problem here. Okay. So to conclude, the idea I'm trying to push forward is that these tolerance landscapes and the thermal death time curves, they should be adopted as the common currency to measure thermal tolerance, right? Many people ask me, so what about the models? Like, it's not even the fact that it's a model. This is the way you should measure thermal tolerance or describe thermal. That's the trait. Right? And dynamic models, they provide way more refined predictions than what people have been doing with critical thermal limits. So it can improve the predictive power at multiple scales, right? You've seen it happening within days. We can extrapolate it up to years, months, seasons, you tell me. But the big problem nowadays is that validating the predictions is always a challenge, right? It's difficult to measure mortality in the field, and so many people are skeptical of the potential use of these type of tools because it's difficult to validate. But as I showed you guys, there, in all the systems that we try to validate, we've been able to validate with a very, very high degree of success. So even though it remains urgently necessary, I would encourage everybody to switch and adopt this new paradigm, right? And that's why we have here Europe in low res, and there you go, Europe in way more high res, because I think that's the analogy with what we have with the thermal tolerance landscapes and versus critical thermal limits. Okay, and with that, I'm happy to answer questions. Thank you very much.
Lots of food for thought. Open for questions and discussion. Yeah, Sahar.
I was wondering about the validation, whether in some part you made it against the abundance, or was it always against, in the field, was it always against like the mortality itself?
It depends. So that's why nowadays when I show this presentation, I show that there are many different ways of validating it, right? So for example, in this capture-recapture experiment here, this is mortality.
Okay.
Right, so we have one individual per vial, and we release these individuals, so you can see the cage there in the river, and we would go every day and collect and see which ones were alive and which ones were dead. So in this case, it's really mortality,
Yeah.
and we really have the temperature measured at that particular environment. All right. So in this case, you can do mortality. You have to be creative because you have to recapture the organism, but it's feasible. So in this case, it's mortality in the strict sense. In here, this is a relative abundance.
Okay.
Right? So again, it depends on the system that you're working with. You could try to do it with laboratory conditions, natural, semi-natural conditions. It's up to you.
Yeah.
But can you validate it with mortality? In the field, ideally, sure. It's just difficult, but
Yeah.
you could.
And I was also wondering, how much do you think these thermal limits data is available for marine fish species? For how many fish species can we have this data now?
Yeah, so...
And is it accessible?
Yeah, if you take a look at this paper now, it's just in evolution right now. So I don't remember. So in this case, the validation, we were able to cross-validate, if you want, the 21 species that you had in common in one review versus the other review. All right. Now, for thermal death time curves, if I remember properly, for fish species in North America, we already have something like 250 curves for 52 species. Right? So one of the things that happened was, by the time we realized that this was important, we realized that depending on the area, fish physiology in particular, they have done a lot of work already. And many of them were published before the 50s. So we had to go into this kind of lost literature, right? And there's a lot of data there. So now I have a PhD student, and he's doing this review of, and the other thing is, let me see if I can show you here. Oh, where's the, this. And we were also able to show these are endpoint data, so these are critical thermal limits. In many cases, you can see the ramping there, all right? But we were able to show that, with some, as I said, statistical hocus pocus, you can actually reconstruct from these data, if you have enough data, you can reconstruct the function, okay? It's not easy, but it's feasible. So what he's doing now is he's compiling everything that was published. I mean, by now I think we have 10,000 records or a little bit more. And we're going to combine the curves with this empirical data here, and we want to reconstruct how the thermal death time curves look across the tree of life. And then we want to see if it has phylogenetic signal. Can you use something for closely related species? That's, we don't know. But there's more and more data available. And there's a lot in different sources of literature as well. For instance, bacteria and fungi, there's a lot, but in the food processing literature. Nothing to do with ecology, but they have a lot of data.
Thank you. I had the exact same question with Sahar in the first part, whether it was abundance, whether the studies that we see are based on individual or abundance-based protocols, coupled with the question, what do you think about the reproduction rates? Do you think when we have an increase, maybe an increase of temperature, maybe we have also an increase in reproductive rates, and we can somehow have a compensation of the loss in community level?
It's a good question. It's a difficult question. So by the time the model came out, there are many, it's a black box in the sense that, we know it works well for mortality because we understand how mortality works. So that's fine. But then many people say, that in principle, you should be able to use the same landscapes, or this region here, to calculate to some extent sublethal effects, because sublethal effects must be, by definition, confined within whatever is lethal. Right? So that means, for instance, that reproduction has to be happening or being able to occur within the white area. It cannot possibly occur in the gray area. Right? So sublethal effects of temperature, they should be confined within these limits here. Now, how that works, I don't know. It's difficult to measure. All right, and you would have eventually this problem that, for instance, reproductive rates, they might increase thermodynamically because of temperature, but eventually they will end up reaching this limit as well. So one of the things that we'll be talking about in Granada with some other colleagues is how you can actually try to generalize this model, because this is mortality as a function of temperature, but we also have performance as a function of temperature, and put that in multivariate scale. So you can actually, it becomes a volume. Okay, and we have some ideas, but it's very difficult to parameterize this sort of model. Very difficult.
More questions? Comments?
Thank you for the talk. It was super interesting, and it really shows very strongly the importance of physiology. But I wonder if you have an idea of the importance of biotic interactions in all this, and whether you can create these landscapes across trophic levels and you see a difference or something like
That's
this.
the idea. Yeah, so there are different ways of approaching this issue. So we wanted to include, and it's not in this presentation in particular, but I had another presentation where we wanted to combine the landscapes and estimate them from different nodes and see what might happen with, a collapse of trophic systems and food networks or pollination networks. The problem right now is that it's difficult to have the thermal tolerance measured for every single node. But we wanted to see something more general. For instance, we would expect, that was as a rule of thumb, that producers would have a higher tolerance than primary consumers and then secondary consumers. You would expect that if you think that these communities, they have been shaped by natural selection in the long term, right? Because you might have the collapse of a top predator, but then there's no cascading effect. But if you have the death of a primary producer or a keystone species, everything else would collapse, right? So based on that knowledge, you'd expect that primary producers will exhibit a higher thermal tolerance than primary and so forth. We looked for that. We never found evidence for that. All right. So the question is, well, communities, they're not closed systems. It's like we're working with this, assumption that essentially what we see here is what has been shaped by natural selection in the long term, but you have a lot of colonization, migration, and so forth. But I know that there are many people trying to combine both. And it's not trivial, because it's difficult to measure thermal tolerance, and networks, you need information from many different nodes. So I haven't seen a network that is highly resolved with a lot of species, and that we have the thermal tolerance for most of them. We don't.
I have... Oh, okay.
Hi, really nice talk. I have one question. Have you talked about animals that physiological can down on of regulate metabolism to control for these changes? I ask this because I found that I found other species that use diapause, the winter diapause mechanisms to control summer extremes. So that will be, the species was able to use different physiological mechanisms to control for this, so they can regulate this, so the models can be applied in that area.
Yeah. So we've done, for instance, there was a paper in 2018 where we measured the curves, but for different developmental stages, and it changes dramatically. So larvae, they could tolerate, I don't remember right now, but I think larvae could tolerate more of an acute, so in the short term, but then the slope would decrease very rapidly. And as they grew up, the adults, it was the opposite. They couldn't tolerate so much the acute, but then it had a resistance to a long-term exposure. So it does change. So, that's a very good point, because when I mentioned that's thermal tolerance, the trait, it's not a fixed trait, right? I mean, the trait, just like metabolism, in principle, you should visualize these landscapes varying all the time. So if you go and you have warm acclimation, the whole curve is going to change, right? And one of the problems, and it's a big matter of discussion in the literature, is that by the time you're measuring the trait, the animal could be acclimating as it's being measured, right? And so you have the problem now that even though what we have is a theoretical approximation, and it seems to work quite well, of course you have a problem by the time you're measuring something, you're altering the system and it's going to respond to the measurement conditions. So the animals, they might be acclimating to what you're measuring in the first place. It could be happening, all right? So again, that's why I say it's the trait, right? And the trait might be going up and down. The landscape could be changing all the time in real time. And it's going to be changing depending on what physiological mechanisms you're working at the time. Diapause insects are going to have a very different thermal tolerance than those that are active and so forth. All right? But in principle, you could measure them. If you're creative enough and you have enough resources, you could measure the sort of plasticity in this type of trait.
So if I have the data of survival, we can model that.
If you have the data of survival, I don't know. If you have the data of thermal tolerance, you
survival.
might.
Okay.
You might, yeah.
Hi, thanks for the talk, very interesting. And also related to this past question, I mean, this kind of acclimatization or plasticity you were mentioning must have some kind of cost. So I was wondering whether there are any studies or you're planning on looking at generational effects, maybe?
That's a good question. So many people say that physiological plasticity has costs, and when you go and try to measure it, it's difficult, right? And with regards to heat tolerance in particular, I think the reason why people haven't seen it so much is because you have critical thermal limits having so much noise that it might be difficult to quantify any type of cost, okay? Now, we haven't done this for thermal death time curves as it is right now. We're just publishing, there was a journal of animal ecology that was accepted the day before yesterday, and so it's coming, I don't know, online maybe in a couple of weeks, where we tried to see the limits of plasticity. So we acclimated Drosophila between, I think it was 15 up to 30 degrees, seeing what might happen with the curves as, you know, as acclimation temperatures increase. And we can see some type of limit. Now, we couldn't see any type of, you know, explicit costs. So we don't know if there are costs there. They should, in principle, there should be, right? I mean, but we haven't seen empirically any type of cost. Oh, transgenerational plasticity is tough. I mean, that's another subject. I have my problems with transgenerational plasticity, but we can talk about this, yeah. But as far as I can tell, nobody has done anything in the source of transgenerational or even heritability. We're trying to publish heritability for these curves because it's also difficult to obtain it, right? Because it's no longer a value, it's a function. And calculating heritability of a function is very tricky. So we're also trying to work on that. And then if we can measure heritability of this function, we could be able to get something like transgenerational plasticity. Yeah.
So I'll have a couple of questions. One is more conceptual and the other one is practical. Starting with the conceptual one, so those that were influenced by the work of Hutchinson and the definition of fundamental niche versus realized niche, with the fundamental niche being, for example, applied to thermal limits, the thermal limits of organisms without interaction with environments, right? What would be inherited, like the characteristic of the organism, what would be what we talk about, what we mention as a trait, right? And then it would be the realized niche, which is the interaction between the fundamental niche and the environment. So that's the way, and that is not a trait, right? That is the interaction between, but according to your formalization, in a way, what you're doing is a kind of a realized niche, right? Because you're looking at how the physiology interacts with the environment through acclimation and generating a landscape, which would be a mechanistic approximation to a realized niche in a way, and you're calling it a trait.
Yeah.
So in a way, that kind of challenges the established view of what a trait is and how you measure it. You know, I'll be interested to learn about your thoughts about that, and then I'll make the second question.
If I have to make a definition regarding what we did here, I would say that this is some way of approaching the fundamental niche, right? I mean, it's an abiotic environment, it's temperature. The problem is that in the concept, or the historical concept of fundamental niche, you think of, you know, the environment as a fixed entity, right? It's a temperature, and there's boundaries. But the problem is that the boundary is actually the temperature is increasing, decreasing all the time, and the animal, its phenotype, is also increasing and decreasing all the time. So that's what I talk about conceptualizing things as a more dynamic thing. So you can imagine, for example, you have mammals that they don't reproduce during winter, but they do reproduce during summer. So the question is, what's the real niche there? And it's like, well, it depends. What are you using as the boundaries? Are you using, annual temperature, mean annual temperature? And so I think this is a way of working with a fundamental niche, but you're including a temporal variable in there. There's always time. And it's something that I think it has been very challenging for biologists in general. Is that we always try to use a very static description of reality because it's more convenient. But the real reality, if you will, right? It's animals that respond, organisms, they're living creatures. They're responding to the environment every single time. In order to remain alive in 10 seconds, they have to be alive right now. Right? And the same applies for lineages. In order for the lineage to persist for 2 generations, it has to remain alive right now, and it has to have offspring, within a couple of days. And so how this organism interacts with its environment, in my perception this year, it's a very, let's say, a very explicit visualization of how the fundamental niche operates, just in the temperature axis, right? But I think ultimately it's fundamental niche, not realized one. But then the problem is that the fundamental, as I said, it's not a hard boundary. The boundary is changing all the time.
Or maybe the fundamental niche doesn't exist, right?
Could be.
And the realized niche and the fundamental niche are just constructions. What exists is the niche. And we try to approach it in different ways.
Yeah,
maybe.
Our models and our conceptualizations are useful until they collapse. Right, and so I think it's a useful abstraction. It helps us to, compare tropical versus, temperate organisms. It works fine, but one of the problems and one of the challenges that is underlying this is that all this framework worked very well when we wanted to compare organisms from a tropical environment versus, an organism from a cold environment. But then when we needed to predict, we realized how poorly our conceptual model was, right? And so it was pretty bad, and then we needed something better. But it remains an abstraction, right? It remains an abstraction.
So my practical question is, a few years ago when I was doing this sort of work in physiological work, and of course the holy grail was to try to use physiological models to predict the species distribution on the ground, as opposed to the envelope models that you also mentioned. And we bumped into a practical difficulty, which is the envelope models, what they measure is the exposure of organisms to ambient temperature, right? That's how you calibrate your models. You have locations where the species are, you have ambient variables, temperature, precipitation, etc. You don't really know what's the body temperature, right? But the physiological models, they're based on body temperature, not ambient temperature. And that connection between the body temperature of an organism and the ambient temperature, although you can use some equations that kind of translate it through operative temperature, they're usually very bad. Not least because the organisms, they adapt, they move, they behave, and they don't let them have that operative temperature as if they didn't, as if they were not alive. So in practice, that translation between the physiological model to a biogeographical model, we found we just didn't manage to do it properly. So what I thought about it, because you seem to have given that step.
No, absolutely, I agree. I agree. When I was mentioning, let me see if I have the slide here, and I was talking to the students yesterday. So one of the problems that we have here, right, I was mentioning this, there you go, this slide, right? Oh, the slide's there, yeah. So the problem is that when we say that physiology predicts ecology, that might be the case if you're working with all the variables at hand and they're, correct if you want. So for example, the model, it translates temperature, right, into mortality. You better have the correct values of temperature, right? So if it's not the temperature of the microclimate or, whatever, the animal might, hide from the sun and so forth, and you're using, temperature values that are incorrect, you're not taking into account behavior, the predictions are going to be incorrect as well. Right? And the same applies here. For instance, many people trying to work with metabolic rates to measure things like, whether they could coexist in a given environment or if they can thermoregulate and so forth. But the problem is that whatever you measure in a metabolic chamber, it might not reflect exactly what the animal's, perceiving in the field or how it works. And so the big leap that you have here from physiology to ecology is that there is a theoretical leap here, that you need to somehow account for that. And one of the errors that we did, one of the mistakes that we did, for instance, with critical thermal limits is that, right? You measure it and you say the animal in this chamber, in this water bath, it tolerates 40 degrees. And therefore, in the field, it should tolerate 40 degrees. No, you're not doing the leap, right? You're simply translating whatever you get in the lab to the field without understanding exactly how the organism interacts with its environment. So I agree with you. There's a problem with that. I mean, ideally, if you have some physiological understanding and it works properly, you can come up with some decent predictions. I mean, I'm amazed that this works so well. I never expected it would work so well. But it's temperature, right? We're going to the extreme temperatures, right? So it's not interacting with any organisms. I don't know what might happen, sublethal effects and so forth. But so far so good. I mean, we at least we have one example where physiology does some pretty decent work in the case of predicting what might happen in ecology. But is that a general case? Are we going to be able to do this all the time? It's up to debate. It's up to debate.
Okay, we have space for another last question.
Thank you for the talk. Very interesting. I have a question regarding the end of the talk. Could it be possible that the photosynthetic collapse with temperature could be related, could be a cause of the recent increase in forest fires we've had?
Oh, I would say no. I would say that's dehydration. So it's actually the trees and the plants in the forest, they have less and less, well, it's a combination between lack of water and then increased temperatures, and then you have more fires. But one of the questions that, so people are seeing that you have more and more droughts, and one of the big questions that many plant physiologists or forest ecologists have is why sometimes you have droughts and with the same thermal regime and the same water regimes, and you have a forest that can survive and tolerate that, and then 2 months later you have the same thing and the whole forest collapses, the whole forest dies. And nobody really knows. And so one thing is that you have an accumulative effect, it's accumulated, but people don't know exactly what's accumulating. And so one possibility is that you might have something like a photosynthetic collapse, and then the plants, essentially you might have, let's say, 50 or 90% of the leaves, they're collapsing, and then the plants can no longer remain hydrated, and eventually the individual dies. But plants are tricky, huh? I mean, it's difficult to make predictions with trees.
Okay, thank you, Enrique. Let's put
Thank you
our
very
hands
much.
together to thank Enrique for...